All right, folks, let's get started. So it's a very great pleasure for me to introduce Jun Huh today to tell us about his work on neuroimmune interactions. So Jun is Associate Professor of Immunology at Harvard Medical School. And Jun is one of the young leaders in the old as well as new and emerging field of neuroimmune interactions in development and in understanding both normal function as well as abnormal function in brain disorders. Jun did his Ph.D. at the California Institute of Technology, or Caltech, in 2005. Then he was a postdoctoral fellow at NYU School of Medicine with Dan Littman. And then in 2013, he joined UMass Medical Center, Worcester, as Assistant Professor of Immunology. In 2017, Jun moved to Harvard Medical School, where he's now Associate Professor of Immunology. Jun has really become known and will be even more known, and you heard it here first, in pioneering studies elucidating mechanisms by which the peripheral immune system influences brain function using cytokines as modulators. So in a series of beautiful studies, Jun has shown how maternal immune activation can lead to cortical and behavioral abnormalities via the effector of Th17 cells through IL-17. And together with our own Gloria Choi, Jun has shown that how in genetic mouse models of autism as well as maternal immune activation models, how inflammation-dependent rescue of behavioral deficits, recapitulating the finding that in some ASD children, fever reduces symptoms. So Jun and Gloria have come up with the brilliant idea that cytokines, certain cytokines based on certain genetic or environmental backgrounds can have ameliorating effects on behavior via various neural mechanisms. So I'm not going to steal all of Jun's thunder, but it's been wonderful to have Jun as part of our community. His work has influenced our own work in many ways, and we look forward to hearing him talk about neuroimmune interaction underlies neurodevelopmental and immune phenotype.
Thank you. Does it work? Okay. Thank you so much for a very kind introduction, Mriganka. It's such a pleasure and honor for me to present our work, here at this MIT, because I feel like MIT is my second home. So today I'm going to tell you about one of our recent study, but I will try not to show many facts plot or both with or the immunophenotype, but try to tell you a core finding of our work. So I was thinking about how to start my talk, but I decided to show you a few graphs as an introduction. So these are the incidence rate. Graph on the left show you that those case number for many different infectious diseases has significantly decreased over the years. So as a society, right, we have done pretty good job in keeping this number down. Perhaps until we are hit by COVID-19. But graph on the right, gave us a very different, picture. This is also, prevalence rate of those immune-related disorder. And then, as you can see, unlike the graph on the left, the case number has been, on the rise, and it's not clear yet, what drives such, rapid increase, right? Many of these genetic immune disorder, has a genetic contribution. People have identified some, through GWAS or other ways, they identify some genetic mutation that contribute or increase susceptibility of developing this immune disorder, but they have not identified the major genetic mutation, right? So this data suggests that, right, perhaps, non-genetic factor, environmental factor, you can call it that way, may also contribute to, this, immune-related disorder, right? So let me show you another graph. This is also incidence rate, of, neurodevelopmental disorder, autism. So as you can see, according to CDC, this is, I have to admit that this is a little outdated. So as of 2022, according to CDC, every child, out of like 46, 48, children born in the States, diagnosed with autism, right? Again, it's not clear, what drives such, rapid increase, right? Like there's a lot of, thought and hypothesis, but perhaps, better awareness, right? Or better diagnostics, may have contributed to this rapid increase, right? But, one thing that's abundantly clear to me is that, this kind of rapid increase cannot be explained by genetic mutation alone, right? Because it's very hard for me to imagine that we have so many genetic mutations over the course of 20 years. So this data really suggests that perhaps just like an immune disorder, like a neurodevelopmental disorder such as autism also, may be dependent on other non-genetic factor or environmental factors, right? But when people say environmental factor, what they are, how they work, it's not really clear, right? So one of the things that my lab is focusing on, along with the Gloria's lab, we've been trying to understand, how this, non-genetic factor influence brain function and, brain development in the context of a neurological disorder. And the one environmental factor, actually, we are focusing on two different environmental factors, but one factor that we have, initially focusing on is the gut microbe, right? Bacteria in and on our body. So we've been able to show that bacteria, microbe in the gut, have ability to control or modulate the function of our immune cells. And more recently, we are able to show that these bacteria, take host-derived factors such as bile acid, turn those bile acid into immune modulatory bile acid metabolite. In doing so, these bacteria has the ability to control function of inflammatory immune cell and also anti-inflammatory immune cells. I don't have time to show you those data. But also with the Gloria, we've been also able to show that peripheral immune cells have ability to control brain function and development. And then, we studied this interaction in the context of autism, and also more recently, we started to look at this, assess this interaction in neurodegenerative disorders such as, AD or ALS. So just for this audience who do not think about immune cells every day, I just prepared the next slide. So I personally find that there's a lot of commonality between the immune system and the nervous system, right? They are both very good at receiving signal from this outside, right? Just like neurons, immune cells express, many different types of receptor, and that's what they were evolved for. And that they also very good at, sending up those signal and then communicate with, other cell type, right? Immune cells, very well communicate with a non-immune cell, and just like a neuron does. However, there's also a unique aspect of immune cell. They are not physically, restricted, unlike neurons. So they can migrate from one place to the other. So like one example is that, when you may have heard about this term called gut-brain axis, right? But gut and brain, is separated. But like one idea that we are actively pursuing is that immune cells, receive input from the gut, migrate into the, proximity of the brain, and by sending up their signal in the form of cytokines or other soluble factor, they are actively communicating with cells in the brain. So that's what we are studying. So both the immune and the nervous system, they receive signals, compute them, and then sending up those signals. In doing so, they can both function as a conduit for information flow. So that's how we view this. And then obviously both immune and this neurobiology field has been been there for many years in each kind of separate world. But nowadays, more and more recognition out there that these two systems actually work together to maintain homeostatic balance. And when this, close, relationship or interaction is dysregulated, there could be incidence of, immunological disorder and neurological disorder. So we want to study that. And so another environmental factor or non-genetic factor that my lab is actively focusing on is this, pregnancy. As you can imagine, the pregnancy poses very interesting challenge, particularly for the maternal immune system. Because if the immune system is get too activated, like too stimulated, then it may lead to the fetal rejection, right? So the fetus is essentially is a foreign body to the maternal immune system. So their activity need to be tightly regulated. On the other hand, maternal immune system they need to protect both pregnant woman and also developing fetuses from invading pathogens. So again, they need to find this fine balance. When this regulation doesn't work properly, then there will be long-lasting effect on offspring's health. Like one of the examples that I show you here is that this neurodevelopmental disorder such as autism. Like many studies around the world have shown that over and over again that when this fetus is exposed to uncontrolled inflammation during particular time point of pregnancy, such as the late first trimester or second trimester of the pregnancy, they have a higher chance to develop neurodevelopmental disorder such as autism, right? So understanding this process, right? I'm not saying that pregnancy is the only environmental factor that may contribute to to this increased chance of autism, but this could be one such a factor. And then when we think that studying this process is very important, particularly as we live through this COVID era. Because if you look at, again, according to CDC, as of July 2022, more than 200,000 pregnant women has been exposed to SARS-CoV-2 induced inflammation. So when people talk about this long-term COVID effect, like long howler phenotype, brain fog, but another like a long-term COVID effect that we think that is coming is this, this like long-lasting consequences of exposure to COVID infection on this offspring. So we think that there will be increase in the autism case number, and also other increase in the also immunological disorder in the future as well. So we need to study this process. And then, thankfully, the scientists previously, including Paul Patterson, has established this rodent model. This is called the maternal immune activation model. And people essentially are using different types of immune stimuli. They use polyacid to model viral immune induced inflammation or LPS or other bacteria or viral infection. And then they found that this mouse offspring in their adulthood who are exposed to this inflammation during pregnancy, they display these changes in their behavior and also neurodevelopmental phenotype. Of course, we cannot be sure that these mice are autistic. Just we cannot be sure with monogenic mouse models as well. But what we can know for sure is that this, this as a result of early exposure to the inflammation, many months after, there are changes in this, their animal behavior in the brain, neurodevelopment. And using this model, we want to understand, how this process, takes place, right? So, using this MI model and also genetic tools and the various, immunological and neurobiological technique, we were able to identify the key players in this process over the years. So as Mriganka introduced, we are able to identify maternal Th17 cell. This is a type of T cell that produces various cytokines, including IL-17. And then these Th17 cells, we found that they play important role in promoting this behavior abnormality and other neurodevelopmental phenotype in the offspring. Mechanistically, we show that this Th17 cell produce interleukin 17A or IL-17, and they cross the placenta barrier. And then they act on this receptor that expresses in the nestin positive cells in the CNS of the developing fetuses. If we remove those receptors in the fetuses or remove Th17 cell in the mother, then genetically, then we can prevent the development of this behavior abnormality. Once this receptor is activated, that led to this activation of integrated stress response pathway in the male, but not in female brain. We don't know yet what drives such gender sex differences. But this increased level of ISR contribute to this phenotype. If we block this ISR pathway by giving them with a chemical inhibitor or genetically block this arm, that we can prevent this phenotype. And then also, related to the later part of my talk, we are able to show that the type of gut bacteria this pregnant mice has really make a huge difference, because we show that the same type of inflammation is given to pregnant mice that do not have this one particular type of commensal bacteria that promote Th17 differentiation, then that inflammation is not sufficient to induce behavioral abnormality in the offspring. Only when these pregnant mice carry this bacteria, promoting Th17 cell, then these mice become susceptible to inflammation, and then their offspring develop this behavioral abnormality. So this is one nice example where three environmental factor, like inflammation, maternal inflammation, type of bacteria in the gut, and pregnancy work together. And when those three factors, happen to coexist, then this offspring develop this neurodevelopmental phenotype, right? So humans got to be much more complex than this. And then after that, so we, like many other factors, IL-17 in the pregnancy, promote the behavior abnormality, but in the same molecule in the different context, as Gloria showed in 2020, this IL-17 can function as a neuromodulator and act on this pre-existing circuit, and actually in the adult mice, it actually rescue or mitigate this behavior abnormality. So enhance sociability phenotype. So this is a really interesting, giving us exciting avenue to further study this process. But basically, telling us how complicated this circuit could be, and then how how powerful this immune system can influence both brain function development during the pregnancy, and the function in the adult mouse offspring. So the pregnancy, I told you that this offspring exposed to environmental factor inflammation during pregnancy can develop this neurodevelopmental phenotype, right? But data also suggested that not just a neurodevelopmental phenotype, but also immune-related phenotype also can happen in this offspring who are exposed to various type of environmental factor during pregnancy, right? I mean, this is really interesting to us, and then it makes sense, right? Because during pregnancy, many things happen, right? It's 21 days in mice, 40 weeks in human, right? There's like organogenesis, neuromigration, so many things happen. But what do we understand about this pregnancy, right? It's really, I think it's one neglected area, and I try to, whenever I have opportunity, I try to argue that we need to study more about the pregnancy. What happened during pregnancy? How they influence maternal health, and also how that also influence fetal development, right? So in my lab, we are also studying the long-lasting effect of a pregnancy in the immunological context, and also studying what happened the first time pregnancy influence the second time pregnancy. So there are so many questions that we can ask. So this is the idea that due to the exposure during pregnancy, that these children develop this immune-related phenotype, right? This is interesting because, in the context of autism, again, as Mriganka said, earlier, that, some autistic children often display this immune-related phenotype as comorbid symptoms, right? It's a very different, phenotype, right? Neurodevelopmental phenotype and immune-related phenotype. Why do they have both phenotype, right? Why do these, seemingly very, different phenotype co-arise? And if, according to at least one paper, this paper suggests that, these autistic children even have a higher chance to develop, inflammatory bowel disease, right? This is IBD, compared to, this, control group, right? So the question is how these two phenotypes co-arise. And then also, if you look at this in autistic children, not all of them, but small percentage of those autistic children, tend to have this primed immune responses. So they have, compared to healthy control, this small group number of autistic children tend to have an increased level of cytokine in their blood circulation. Okay, and then when you take the immune cell, because it's a nice thing about the immunology, right? You can take immune cells out and put them on the dish and then stimulate them in vitro. And then they found that those immune cell monocytes from autistic children tend to produce more cytokine than immune cells collected from control group. So they show this in immune primed phenotype. And also T cells, when they take a T cell, not the monocyte, and put them in the vitro and stimulate them, those T cells from autistic patients also produce higher level of cytokine, right? So the question that we are asking is, how, whether this MI mice, exposed to inflammation during pregnancy, whether they also show immune primed phenotype in addition to, abnormal behavior phenotype, right? That's what we wanted to ask. But we already sort of know the answer even before we start this work, because, earlier papers, from one from Gloria's lab, and then also another from, Paul Patterson's lab, already show that, this, compared to control group, if you look at this, MI mice in this case were challenged, with, LPS, they produce more cytokine in their blood circulation, right? So it's just like a, human, they have a higher level of cytokine in their circulation, and then also immune cells when they collected from MI offspring compared to PBS offspring and stimulate them in vitro, they produce more cytokine, right? IL-6, IL-17, as a readout for inflammation. So yes, they seem to, have this immune primed phenotype, but we want to understand the mechanism. And before we do that, we first want to look at their baseline immune, profile, right? PBS offspring, MI offspring, and then, we just look at, this is a busy slide. You don't need to read these graphs, but what you can see is that they are not different, right? So when we look at their baseline immune profile, control group, MI offspring, same, right? So we decided then to take these MI mice and then challenge them again later in their life with another immune stimuli. So again, just to show you again, the only difference is that we gave one injection at E12.5, embryonic day 12.5 to the pregnant group, and then so PBS offspring and MI offspring, and then we waited until they become adult, and then we re-challenged them. In this case, we basically gavage them with pathogenic bacteria, gut bacteria, so to induce gut inflammation, okay? So there's all the technical detail you don't need to worry about, but so then we look at this, their gut, like inflammation, right? So the shown here is a colon, and then the shortened colon length means that it's more severe inflammation. So control group, we use just a little bit of a pathogenic bacteria. So by itself, it didn't induce a very strong inflammation. But if we do that introduce that bacteria into MI mice, their colon gets shortened, right? Meaning that they may have a more severe display, more severe inflammation. Okay, and then when we look at their histology score, we found that they have they display more heightened inflammation. Okay. So it's a cell death, it's a kind of common way to initially measure the degree of inflammation. I mean, very, very strong, under inflammatory condition, then their colon gets shortened. Yeah. And then also we systematically, we look at the different level of cytokine, and then, following this gut infection, okay? And we saw this increased level of cytokine in the MI offspring compared to PBS offspring. We also want to look at, immune cell, but, don't look at this FACS plot. Just, let's just look at this, graph. But basically increased level of, immune cells in this MI offspring. This is from the colon, okay? The site of inflammation. So we detected, increased level of inflammatory immune cell, and then decreased level of anti-inflammatory immune cell, right? It's like, we have like a less level, reduced level of inhibitory neuron, more activating, excitatory neurons. So okay, so these mice show this enhanced inflammatory phenotype against this one pathogen, right? So we want to ask whether this finding can be generalized. So we decided to use a few different immune stimuli. Use a few different immune stimuli. So this is anti-CD3 inflammation. So this is basically a way to induce a massive T cell activation. And then we found that this compared to PBS offspring, MI offspring, I found with the increased level of T cell upon this challenge. Okay, and then just next model we use, this is a brain inflammation. It's like a MS, multiple sclerosis mouse model. And when we did that, this MI offspring show more severe phenotype. Their phenotype is worsened compared to control group. So it doesn't matter. We use 3 different models, and we found that this MI offspring show this enhanced inflammatory responses against stimulus. However, not all the stimuli work in the same way, because when we use a DSS, this is a mouse model to induce wounding through the to the colon. It's more like an innate immune-dependent model. And then when we did that, we don't see the differences. Models above is more T-cell dependent model. Okay, so when we did a bunch of this kind of immune challenge, right, we are able to not only find that this MI offspring show this more severe inflammatory phenotype, but this phenotype may be related to T-cell, not innate immune cells. So that's how we identified. So the answer to the first question is, does the MI mice, show this inflammatory phenotype? The answer is yes. And the question is, what underlies this phenotype, right? And so, when we do not even have a hypothesis where to start, right? We often wanted to identify context, where these things, take place. So that's why we decided to, perform this, cross-fostering experiment. Okay, so mice were, pregnant mice were induced, vehicle versus poly-IC, right? And then waited until they, produce pups, right? And as long as they produce pups, postdoctoral fellow, she basically switched the mother, right? So this is basically MI mother, PBS mother, and then it's offspring. So it's a 2x2, 4 different conditions. Okay. So previously we did a similar experiment, and then we're able to find that this neurodevelopmental phenotype, behavior abnormality, was determined prenatally, right? The reason why I say this is, we reproduce this phenotype. So compared to control, PBS offspring, MI offspring, show this social deficit, and also show this increased, repetitive behavior, phenotype. This social deficit, this is 3-chamber assay. This social deficit, still, observed in this MI offspring. The mice born to MI mother exposed to inflammation, but raised by control mother, they still show this social abnormality phenotype. If you look at the second column, right? And then likewise, control offspring, even though they were raised by MI mother, they still show good sociability phenotype, suggesting that this behavior abnormality, was determined prenatally, right? Then what about the immune primed phenotype, right? So I told you that, we use these various methods, and then, look at using, here in 4 different readout to capture this immune primed phenotype. Let's just focus on one. This is an increased level of, Th17 cell in the colon of mice that exposed to this, bacteria infection, Citrobacter infection, right? In this case, if you compare control group versus, MI group, there's an increased level of Th17 cell in the MI group, right? But this is getting interesting because, when you look at this MI offspring, mice born to MI mother, raised by control mother, right? But they no longer show this, increased Th17 cell phenotype, meaning that this immune primed phenotype disappear if we switch, birth mother with the foster mother. Okay. The third column is contrarily this PBS offspring, right? The mice born to control mother when they are raised by MI mother, right? Mother exposed to inflammation during pregnancy, now they start to show this immune primed phenotype, increased level of Th17 T cell. Okay, so unlike this, behavior abnormality, neurodevelopmental phenotype, this immune primed phenotype seems to be determined postnatally. Okay, please ask me a question if it's not clear. So when you talk about this postnatal effect, right? Something that happen after birth, right? By and large, you can think about two possibilities, right? One is milk, right? So maternal milk somehow influence this phenotype, and second is microbiome. Okay, because essentially during pregnancy, we as well as mice, they were kept under germ-free condition in the mother, right? So there's no bacteria there. And then as soon as we, born, right? Get to this world, right? We often say that the first person or first thing we meet is our mom. But in reality, I would argue that it's actually microbes, right? There's tons of microbes, around here. And then the first time we were born, get to this world, there will be a lot of, the stimulus, right? That act on, all this like TLR signaling pathway, immune-related receptor that stimulate, a lot of things that makes, So I think this exposure to bacteria is really need to, study it, like systematically to understand this process. So the question is, whether this immune primed phenotype that seems to be determined postnatally is determined by this or influenced by maternal gut bacteria, right? So before we test that, we first want to know whether MI mice, right, this pregnant mice exposed to inflammation has different types of bacteria to begin with compared to control. So we did, we got the stool sample and did 16S sequencing. So this is a very simple way. Basically, by sequencing this conserved ribosomal region of a prokaryote, because their ribosome is different from eukaryote, we can, gain some understanding about the relative community, gut bacteria community, right? And again, just two different graphs, like, compared to control, we found that once in a pregnant mice exposed to inflammation, and then we collected the stool sample like 6 days after inflammation, that time is sufficient to induce such a massive changes in the overall bacterial community in the mother. And then it doesn't, if we look at the PCR analysis, we see that they're like different segregated, right? Okay. So this MI mother has a difference in their bacterial community, right? But then we want to know whether this changes, alteration in their bacterial community can also functionally, influence this immune primed phenotype in the offspring. So how do we do that? So that's where we start to use this, germ-free facility. So like a mice, generally, we kept them under barriered facility, like a SCSB condition, but they have tons of bacteria and microbes, in and on their body. So this germ-free facility, we keep animals inside this big bubble, so they do not get exposed to any microbes, right, during the development, right? So this is a very reductive way of studying the role of, various microbes on our system. And then, using such, tool set, and then also, by, gaining this ability to monitor animal behavior, while we keeping these mice under germ-free or noroviral condition, by the way, this work was done, through collaboration with Gloria, we are able to study the effect of, microbes, right? In not only in their, on the immune system, but also, brain function and animal behavior, right? So we can look at their, behavior in the germ-free status, or in the condition where we just introduce, one or define the number of, gut bacteria or like skin bacteria, right? And then, moreover, since we have this, reductive system, we can then also introduce, stool sample collected from, autistic patient and AD patient, ALS patient, and then by introducing those stool sample into this, various mouse model for neurological disorder, we can study this interaction between, genetic mutation and also, non-genetic factors such as microbes. So, by taking this approach, right, so we collected stool sample from control mother versus an MI mother, right? I told you that they have difference in their bacterial community. transferred them into germ-free female inside the isolator, and waited until they produce pups, babies. And then we call them as PBS STs to transfer the offspring versus MIs to transfer the offspring, right? In this case, these mice were not exposed to inflammation, okay? They were just born to mother who received stool sample collected from MI versus control mothers, okay? And then we first ask whether they show behavior changes, right? It would have been so amazing, right? Just if a bacterial transfer had to transfer, induce some sort of behavior changes, but the answer is no, right? This bacterial transfer, was not sufficient to make these mice become, less social. They are absolutely social in both cases, right? Okay, so the conclusion here is that, just like our cross-fostering data, this bacterial transfer is not sufficient, or changes in the bacterial community is not sufficient to induce behavior changes, okay? But what about immune-primed phenotype? When we did this kind of experiment in a similar way, we found that this MIS2 transferred offspring now show this immune-primed phenotype, right? Suggesting that, again, consistent with our cross-fostering data, immune-primed phenotype can be postnatally determined in a way that this bacterial transfer alone is sufficient to induce their offspring's immune-primed phenotype. So these data suggest that bacterial community, alteration in the bacterial community play important role in inducing this immune-primed phenotype in the offspring, right? So we want to do this experiment in a reverse way, right? So can we create a condition where we see neurodevelopmental phenotype without detecting this immune-primed phenotype, okay? So how do we do that? So we basically generate MI offspring inside this isolator, right? No bacteria. But we had to, because of technical reason, I told you earlier, you need one bacteria species that produce Th17 in the mother in order to produce MI-associated behavior abnormality. So we generated some monocolonized mice. In these mice, only microbe they have is this called Singleton filamentous bacteria shown here that are strong Th17 inducer. In this condition, this mice only has one bacteria, no other bacteria. We induce inflammation and get their offspring and run behavior. And then show that this MI mice from SAP monoclonal mice start to show this social behavior, social deficit, right, compared to control. What about immune-primed phenotype, okay? We no longer see that. Suggesting that you need some other bacteria, right? Not SFB, to produce this immune-primed phenotype, right? So by taking these various approaches in cross-fostering, SAP monoclonal mice, or stool transfer experiment, we are able to uncouple this immune-primed phenotype from neurodevelopment phenotype in this MI offspring. So the answer to the second question is, what drives the immune-primed phenotype in MI offspring is likely the maternal gut bacteria, right? Changes in the maternal gut bacteria. The third question that we ask is, then what about host side? Which cells, drive this phenotype? I told you earlier, I alluded to that a little bit, that, it's a T-cell related, pathway may be involved, right, for this phenotype. So we decided to just take this immunological trick, like immune system is, they are mobile. So we can take T-cell from PBS versus the MI offspring, and then by adaptively transferring them into T-cell deficient mice, right, we can ask whether these T-cells are sufficient to induce phenotype, right? And then the answer is yes, compared to recipient mice that receive control T-cell, this mice that receive this MI-derived T-cell show more severe inflammation, okay? Suggesting that T-cell, this is not a loss of function experiment. This is showing that T-cells can produce this immune-primed phenotype in the offspring, right? So when we have that data, that will also lead us to ask a more mechanistic question. What happened? Why this T-cell? How do they remember, right? This kind of question. So, to do that, another immunological trick, we can genetically, congenically mark, PBS control T-cell versus MI T-cell using some genetic, polymorphic marker, co-transfer them into same mice, and then retrieve them, and then do the comparative gene expression analysis. And we found that this MI-derived T-cell they tend to express many more genes on the same condition, right? That related to immune pathway, right? Th17 cell IL-6 pathway, okay? So now what I'm telling you is that T cells from MI offspring has the ability to induce immune primed phenotype. They tend to produce a lot more genes relevant to inflammation or immune pathway, right? Then the question is, how do they know? How do they even remember, right? This whole exposure to inflammation happened during pregnancy. Several months later, their T cells still remember what happened during pregnancy, right? So we decided to look at their chromatin structure using ataxic analysis, and then found that this MI T cell have increased accessibility to this transposase, suggesting that their chromatin structures are likely different, and this opened chromatin structure correlate with those genes that involved in the immune pathway. So we still don't know, right? What drive, how do they, what drive this changes in the chromatin structure of this T cell, right? But this data suggests that this exposure during pregnancy, that information somehow stored, okay, or remembered in T cell as a kind of the chromatin structure changes, and then these T cell, when they will get exposed to second stimulus later on, they tend to produce more genes, as a cytokine, or it shows this immune responses, okay? Then, this is the offspring side, and then we next ask, what happens which factor is important, during pregnancy, and so we decided to look at this role of IL-17 because, this maternal IL-17, I told you earlier that it drives this immune, neurodevelopmental phenotype in the offspring, right? So to test whether the same IL-17 during pregnancy can endow this phenotype of this immune primed phenotype in the offspring, we basically, repeated this experiment. This is using PBS offspring, MI offspring. But in this case, we inhibited IL-17 activity in the mother by pre-treating them with antibody, blocking antibody that suppress IL-17 activity, okay? And then so compared to isotype control, we found that this offspring born to mother that were pre-treated with anti-IL-17, even though they are exposed to same degree of inflammation, they no longer showed immune-primed phenotype. Okay? And then so suggesting that this is the maternal IL-17 play an important role here. And then this is again busy slide, but we repeated this is a stool transfer experiment in the presence or absence of anti-IL-17A treatment, and then transfer them into germ-free condition, and found that this maternal, if we block IL-17 activity in the mother, then this offspring received this stool sample, no longer show this immune primed phenotype. Okay, all this data suggested that this maternal IL-17 here play a role in promoting changes in the offspring's T cell chromatin structure, make them more susceptible to produce higher level of cytokine when they get exposed to inflammation. And then the next question we ask is, how does this IL-17, maternal IL-17, induce this phenotype? And then this is maybe a little bit of too much detail, but earlier data suggests that maternal IL-17 can influence the level of antimicrobial peptide production in the gut of the mother. Okay, so we look at this, changes in the antimicrobial peptide production in the gut, and then we saw that there are increased level of certain antimicrobial peptide expression. And then when we look at the effective function of this gut bacteria, this is like a wide, many studied short chain fatty acid is a gut microbial metabolite. We found that this MI mother has a reduced level of short chain fatty acid. So current hypothesis is that since butyrate, short chain fatty acid are known inhibitor of HDAC, so it's chromatin, it can influence this chromatin structure. We thought that perhaps this decreased level of short chain fatty acid in the mother due to the action of IL-17 may promote this behavior, immune primed phenotype in the offspring. So I just want to end with one last slide. So if maternal IL-17 induce changes in gut bacterial community through modulating antimicrobial peptide production, the question we ask is what happens if we remove maternal in the mother, if we remove IL-17 receptor only in the gut, okay, then what happened, right? So that's why we used the Vilinkri. This is a gut epithelial specific crinoline, remove the IL-17 receptor. And then, the data is still very, very preliminary, so we don't have many number. And then when we look at that in the offspring, they no longer show this, immune primed phenotype, okay? So just, what one, just, I think it's maybe very complicated, but just one thing that I want you to remember is that the following, right? When this, during pregnancy, when mother get exposed to inflammation, depending on the type of bacteria they have, there's a, increased, like a level of IL-17. The same cytokine, one cytokine, then can act on this, fetal brain through this receptor in the nestin positive cell, and then that activation leads to the behavior abnormality, such as sociability deficit, right? And however, at the same time, the cytokine can act on different cytokine receptor, in this case, gut epithelial cell of the mother, right? And then that lead to these changes in bacterial community. And such changes in the bacterial community of the mother can influence immune primed phenotype in the offspring. So I hope that I was able to convince you that this, at least in this model, these two different phenotypes, neurodevelopment phenotype and the immune primed phenotype, co-arise because of the action of one cytokine in the mother, but the mechanism of action is different, right? This cytokine action, depending on where they act on, cytokine receptor in the neuron versus cytokine receptor in the maternal gut, there could be different outcome. So I want to thank this big shout out to Jun Huh Kim. She's like a former postdoc fellow in my lab and recently started her own lab in Korea. And all this work was made possible through close collaboration with Gloria. And then if I have 2 more minutes, right, I want to show you just quickly some potential implication, right, of our finding. So I told you earlier that this inflammation in the during pregnancy is bad, in that it promotes autism-like phenotype. But at the same time, IL-17 in the offspring, it can be beneficial, right? I told you about that. And then, Gloria showed that previously that, when she has a MIA versus monogenic mouse model for autism, when they were exposed to inflammation, only MIA, but not the other monogenic mice, show this, social rescue, like mitigation phenotype, right? Because, so basically LPS was a way to induce inflammation, and only MIA showed the social rescue. The other monogenic mice didn't, right? And then, it found that it's likely because these MIA mice, when they get challenged, they produce, higher level of IL-17 because of primed immune responses compared to monogenic mice, right? And then if you bypass this, pathway by directly introducing IL-17 into these monogenic mice, then, you can still, rescue this behavior, phenotype, temporarily rescue this behavior phenotype, I should say that, right? So why do I say all this? Because, I want to go back to this, earlier human observation. As Mriganka mentioned that there is a fever effect, right? The certain group of autistic children, when they were exposed to fever, associated inflammation, because fever is just one, readout for this inflammation. Their phenotype can be, like a core phenotype is, significantly improved, right? And according to this paper, like there will be 17% out of like 2,100 children with autism, 17% of these children responded to fever and display this, improved phenotype, right? So why 17%? What's so special about those children, right? Why only 17%, right? So, if I just a little bit of indulge myself and then, translate, I mean, like, if our finding can be applicable to human, I predict that, those 17% who responded to fever, might have this immune primed phenotype to begin with, As to why they start to, have such immune primed phenotype, I mean, that's something that, we need to study in the future. But I think, those children, it will be super interesting to compare this fever responder versus fever non-responder and see whether they are correlated with this immune primed phenotype. So with that, I want to stop here and then happy to take any question. Thank you. Yeah.
at the time that the
Yeah.
So you don't need a host factor as you showed.
So that MIA mother already has altered the bacterial community, right? In order to produce that, change the bacterial community, you need a host factor.
No, so what I mean, the host factor is in the pups. So you don't need anything changing in pups.
Oh, so these changes in the T cells in the happen, likely happen during postnatal stages. So what happens is, once you start to do the cross fostering, the control mice, as they were raised by this MI mother, they were influenced by this changes in bacterial community, and those changes in altered the bacterial community influence this T cell function by shaping their chromatin structure. Having said that, we haven't looked at their we haven't done an ATAC sequencing with the T cells collected in 1 week, 2 weeks, 3 weeks, 4 weeks after birth. That's something that we want to do, but that's the current hypothesis. Yeah.
But have you tried to inject poly-IC in non-pregnant? Does that change?
So we didn't look at this bacterial community, but previously we look at this IL-17 induction. Even though this non-pregnant mice has the same level of bacteria that produce Th17 cell, we didn't detect this IL-17 in the circulation. So still not clear yet. But something about pregnancy, because when people say, pregnant mother has reduced the weakened the immune system, that's absolutely not the case, right? It all depends. Our recent data suggests that we profiled, immune responses, collected from, non-pregnant, pregnant mother with or without COVID infection, and our data show that T cell function may be altered, weakened to some degree. Monocyte function is actually, improved during pregnancy. So as to how does that happen, I think that's something that we want to understand in the future.
Has anyone looked or tried to administer a neutralizing anti-IL-17A monoclonal to the pregnant dams and see if that blocks or reduces the MIA phenotype?
Yeah, we did that before, because obviously, that's the drug is out there already in the market to treat, psoriatic patient, right? So, I mean, whether or not that anti-IL-17 can be applicable to human pregnant woman who, happen to be exposed to a high degree of inflammation. I think, someday maybe, need to happen, but still, we don't, still a lot of missing information. We don't have a good biomarker, et cetera. So. So we look at, we detected increased level of IL-17 in circulation following Citrobacter infection. At the steady state, we don't see that. And then, we haven't looked at the Th17 cell in the spleen or other tissue. Well, what we want to do is, basically using KDA mice, shine the light and change the color and see whether gut residing T cell go to different places. So that's what we want to do in the MI context, right? To probably get at your question a bit, yeah.
Definitely related to the IL-17 receptor in the brain. Is it clear that there's no IL-17 produced by any cell type in the nervous system, or could be another living receptor?
So at least for IL-17A, so we are not able to detect using qPCR. We are not able to detect using IL-17A reporter mouse line. But that's all we can say. Yeah. So we think it's coming from outside, but yeah, I mean, it's negative data. Yeah.
Do you know what does IL-17 cross blood-brain barrier?
So I mean, so that's we don't, but many, it's actually one of this, again, another unstudied area, right? If you inject mice with the LPS and then do the dye injection followed by IV dye, right? Then you can detect the dye in the brain, right? So when people say brain is immune privileged tissue, and then it's very difficult to get through the BBB, one exception is the immune system. We think there is a reason for that, but there earlier papers show that viral infection also increased brain permeability, like LPS does, but the question is how. So I think that's